Hello, this is Miss Dagenford, and we are going to look at the structure of DNA. The structure of DNA is pretty simple, and the only thing we need to add are some details about the chemistry, which can throw a little bit of a wrench in the works in terms of the simplicity. But the basic idea is not hard. In fact, you'll probably remember this from your honors biology or regular biology classes from your ninth grade or 10th grade. So let's get started. By the end of this video, you should understand the way the base pairing rules operate. You should understand the structure of DNA, the backbone and the ladder. You should understand the nomenclature of the complementary base pairs. You should understand the anti-parallel nature of DNA. We're even going to start on replication, but we'll do a part 2 video on that as well. So when we start looking at DNA, we have these nucleotides. The nucleotides are comprised of phosphate, and notice that they are nice and negative. That's going to come into play with some labs that we're going to do. So the phosphates have an overall negative charge to them. And so the backbone of DNA contains this almost shell of negative charges as it twists around, as the double helix twists. Then we have the nitrogenous base. And then we have the sugar. And the sugar in this case is deoxyribose, hence the name. Now here's where we get into something that we have yet to talk about. Like I don't talk about it in regular biology at ninth grade. Now, if you look at the sugar is actually made of 6 carbons. There would be a carbon here, 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 and here. And in organic chemistry, we number these carbons. And so you'll notice this has little 1, 2, 3, 4, 5. And they're also designated with a little apostrophe, and that's going to be named prime. So this carbon is 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. Now, these numbers end up coming into play in how you can identify the DNA strand, whether or not it's 5 prime, 3 prime. Notice the 5 prime is where it connects to the phosphate, and then the 3 prime on the side, and then the 3 prime also attaches to the phosphate on the top. So that's how you can distinguish them. What's going to be a little bit more important is what we know about the 3 and 5 prime with regards to how DNA can be added or grown, making it longer. When you want to add bases to the DNA, they can only add to this 3 prime. So that's going to be extremely important in how DNA replicates. So we can only add to the 3 prime end, and notice there's this free hydroxyl group there, and we're going to notice that this influences how DNA replicates. Now you'll further observe that a couple of these DNA nucleotides, well, they have these nitrogenous bases. 2 of them only have 1 ring, and then 2 of them have 2 rings. And if we go over here, I've gone ahead and written it down here. Purines have a double ring, and those would be adenine and guanine. Pyrimidines have a single ring, and those would be cytosine and thymine. Now, I've come up over the years with a couple of different ways that you can remember. I just take the purines and then the adenine and guanine and make this silly word purag. It sunk into my brain, and that's just what I used over the years to remember it. The other way students can remember is the purines have a double, and that has a U in it. Pyrimidines, there's the single ring. The I's can help you as well. This can help when you are being asked to apply how the base pairing work, how the shape of the DNA works, how they would be affected with the number of these bases. And then we're going to learn about the base pairing rules, and these work because of the way the hydrogen bonding works. A and T must bond together, and of course this is vice versa. T would bond to A, and they can make two hydrogen bonds. G and C must pair together, and again vice versa, C with G, across DNA strands. And we'll do some practice, but again this should be pretty familiar. You've had this before. And then G and C have 3 hydrogen bonds. So when we put it all together, here is a picture that represents 3 models. We have kind of a schematic model of the DNA helix over on the left that shows really nicely the distance, the base pairing rules, but it doesn't give us much insight into the chemical structure of it. The middle one's really nice because it gives a little bit more information. Now, one thing to notice, this would be 1, 2, 3, 4, 5. And then note here is our 3' and here's our 5'. So overall, what they would declare is that this is the 5' end of the DNA strand on the left. And then down here, 1, 2, 3, 4, 5 on the bottom. And notice the 3' end is the open part. So this would be called the 3'. Over here on the right-hand side, it's a little quirky. Notice it's upside down, and we call this whole idea anti-parallel. One strand is going 5' to 3' this way, and then if we go to the right-hand side, we would have 1, 2, 3 sticking up, and then 4 and 5. And that's going to be true. So same thing down here. We'd have 1, 2, 3 with our hydroxyl group open, 4, 5. And so we'd say this strand is 5' to 3'. So they run in opposite directions. Here would be our A and T with our 2 hydrogen bonds, G and C with 3, C and G with 3, A and T with 2. These are nice hydrogen bonds in the middle. So they are not as strong as the covalent bonds that you would see on the edges. So here and here we'd have covalent. Now that does influence then how DNA can be worked with. DNA, because of the hydrogen bonds, if you have an enzyme that can pull apart the hydrogen bonds, or temperature would pull apart the hydrogen bonds, and the helix can unwind. You're not going to see that with the, it's not going to be as easy with the outside rings, because those are the outside of the ladder, because they're covalent bonds. You would need an enzyme specifically to be able to do that. And of course it does happen, but it's just, it doesn't just separate because of temperature. So here are the details of the purines and pyrimidines. Pyrimidines, notice we've got our nitrogens and hydrogens and oxygens, and these are prime examples of molecules that can make hydrogen bonds. And we notice that, and there's another picture down there. We'll get to that. So just as a nice review, here's our adenine and thymine. They can form 2 hydrogen bonds. Guanine and cytosine can form 3 hydrogen bonds, and it rhymes, GC3. That's an easy way to remember it. How does this affect things? Well, it affects things because notice with 3 hydrogen bonds, these would be slightly stronger. Think about the fact that if you have a Velcro strand that's 4 inches versus a Velcro strand that's only 1 inch, the 4 inches is going to hold more. So you can think of it that way. So here are the hydrogen bonds that can occur. And notice it's forming between the negatively charged either oxygen or nitrogen and the positively charged hydrogen. You wouldn't be asked to memorize these, but you need to have a familiarity if they were to show you a picture of them or asked to kind of apply what you know. And I'll give you an example of this in just a second. One more example just to really kind of hammer that home. And then how do we test it? Well, here's the example I was talking about. One thing that scientists noticed is that if you were to compare a DNA strand, and let's say here we have one now. If this is the one strand, let's start practicing our base pairing rules. What would be the other strand? And I'm going to pause right here for a question for you. And what you should have found is that the other strand would be T, A, T, T, C, G. Let's do that again with another strand. And one thing to notice off the bat, this strand number one, DNA example one, we'd have 1, 2, 3, 4 AT bonds and only 2 GC. And then over here, we'd have the opposite. 1, 2, 3, 4, 5, 6 GC pairs, and then only 1 AT. Now think about how many hydrogen bonds exist. This would be 2, 2, 2, 2, 3, 3. But then if we go over here, notice the overall hydrogen bonds are going to be much larger. So this is going to be stronger. So think about what this is going to do if you heat the DNA strands. If you put them in a hot water bath and the hydrogen bonds start wiggling around, as we know they do with the temperature, think about the fact that this is going to open faster because it's slightly weaker. And we can actually measure these, and it gives us a way to test the overall strength of our DNA strands. And that gives us a sense of how many AT and GC they have. So overall, here's our nice strand of DNA. And I like this picture because it does show you the 5 prime. Here's the 3 prime on the other side. It shows you the anti-parallel nature again. Here's our hydrogen bonds. So when one strand, let's just go ahead and summarize. When one strand is 5 prime to 3 prime, the other complementary strand is 3 prime to 5 prime. The nucleotides can only be added to the 3 prime end. That's the only one that has the open hydroxyl groups. And then the covalent bonds are on the outside. The hydrogen bonds make it inside. So this makes it a perfect candidate for kind of describing the stability of molecules. You have these nice strong covalent bonds on the outside, these covalent bonds that give it this nice steadiness, stability, but then we have these hydrogen bonds that allow the molecule to be opened up. It also then gives us a clue about how DNA is going to replicate. And so if this is our parental strand in the dark blue, if we were to open it up, let's just go ahead and make sure, because they're showing the 2 rings. Let's say this is A, G, A, and then this is going to be, let's just say C and T. The other side should be A, C, T. Now, what that does if we open it up is now exposes the base pairs so that we can bring new ones in. So notice this would be A, C, T, G, A. What would the new strand be? So that'll stop us for today, and then part 2 will be how DNA replicates. Almost.